All right, hello. So today I'm going to walk through a sample transformation efficiency calculation like the one that we did in class. This is an update to the one in class because I made a small mistake when I was doing it at the very end of class. So you could be asked a question kind of like this. I would give you information about a transformation reaction and you would need to calculate the transformation efficiency given the outcome of the experiment. So if we look here, what you can see is that I've provided information about how the reaction was conducted. So here is the volume of the transformation solution initially added to the tube. And then at the end of the experiment, once the heat shock was done, this was additionally added to sort of get the cells to feel better about themselves. So 500 microliters of this and 500 microliters of that and some DNA. Now the stock concentration of this DNA is given here. And then after this whole thing is put through the reaction, and the total volume is roughly 1 mil, 500 microliters plus 500 microliters is 1,000, which is 1 milliliter, and it's really actually 1,003, but if for a test, you can round up to 1,000. And then of this 1 mil, or 1,000 microliters, 100 microliter volumes are put on these 3 plates, and these are the results. So if we're trying to figure out transformation efficiency, we actually want to know 2 things. We want to know how many transformations happened, and that's really the same thing as the number of colonies on a plate. And we're going to divide that by the amount of DNA that was used. We want to know how successful we were, so we want to know how many transformations occurred per microgram DNA. So this first part, I think for most people, is pretty easy. The number of colonies is given on these plates. These plates are all challenged with ampicillin. And this particular experiment was being added as DNA as pGlow. And from your lab, you'll remember that it has this gene called BLA that provides resistance to ampicillin. So cells that receive the plasmid are able to survive on these plates, which otherwise they wouldn't do. And so what we see on all these plates is that, a couple of dozens cells survive and form colonies. Since all of these plates are asking the same question in terms of did you get the plasmid, we can average them. So this top part is going to be the average of these plates. Which is 20. Okay? So we have the first part. Now we need this part. And this is usually the trickier part for most people. So what we have to do is really understand what's happening in the transformation. So what I basically told you is that in the transformation tube, we had a total volume of about 1,000 microliters. It was really 1,003, but I'm rounding down just to make the math a little bit simpler, and you can do that too. 1,000 microliters is the total volume because we added this much calcium chloride, which is a liquid, and this much Lb, which is another liquid, and so we have this total volume. Now, how much DNA went into this whole tube? What was the amount of DNA? Well, we added a volume, so how much DNA in the tube, in the reaction tube? We added 3 microliters, but that's just a volume. That doesn't tell us how much DNA. To figure out how much DNA, we have to multiply this volume by the concentration, which is given as 0.1 micrograms per microliter, which is going to be 0.3 micrograms. So again, think about what concentration means. Concentration means for every microliter, there's 0.1 micrograms. So for 3 microliters, there's 0.1 plus 0.1 plus 0.1, 0.3 micrograms. That's the amount of DNA in this whole tube. So in this whole tube, there's 0.3 micrograms total of DNA. Now this next part tells you how much of this tube right here was actually put onto each of the 3 plates. 100 microliters. So we didn't dump this whole tube on this plate right here to get, if this was the Lb amp, which gave us 17 or 22 colonies, we didn't dump this whole tube on here. So we know that we didn't put 0.3 micrograms of DNA on this plate. How much went on here? We have to know what fraction of this actually went on here. Okay, so the fraction of DNA that was used is going to be the volume that we put on the plate divided by the total volume, which is 0.1. And then we just take our amount, which is the total amount, and we multiply by 0.1 to get 0.03 micrograms. Another way that you can do this is just to take this total here and multiply it by 100 over 1,000 directly. You'll get the same thing. So this is how much DNA went to each one of these plates. And you can double check this by thinking about it logically. If this 0.3 micrograms is what's in a milliliter, then how much would be in 0.1 milliliters, or 100 microliters? It should be a tenth, and that should be 0.03. So now we have all the information we need to actually solve this equation. So we can take our transformation efficiency is 20, right, that we got here, transformations, divided by how much DNA went onto each one of these plates, and that gives us an answer of 666 per microgram DNA. You always want to use those units, of course. So hopefully that was helpful, but what's really helpful is taking this example and putting in different numbers for yourself and then figuring out if you can solve it. So, and that's just, basically kind of practicing for what might you see on a test. If I tell you the volume of the reaction, which is this top line here, and I tell you the concentration of the original DNA, and then I tell you how much was used on each plate and what the answers were in terms of colony production, then you should be able to get to this place following these steps. Whoops, sorry about that. My mouse got a little wild. Hopefully that's helpful. And on Friday of this week, I'm going to be doing another Q&A for the Unknown ID project, but you're welcome to come by with transformation efficiency questions as well. Or just email me any questions as you're working through this problem yourself.